Hi, welcome to this month's webinar within the UK Freshwater Quality Programme. As Finn said, I'm sure if you're a regular, you've heard lots of this before, but we get new people joining all the time. So welcome to those, if this is your first webinar. So basically the Freshwater Quality Programme is funded by Natural Environment Research Council and DEFRA, and it's really to improve our understanding of the sources, behaviour of pollutants within river systems, increase our knowledge of how the pollutants have an impact on the freshwater ecosystem, inform policy, look at ways that we can improve the ecological state of our rivers, and look at better ways to adapt and mitigate risks. So that's the aim of the programme. And within it, there's 5 projects that have been funded. And these are the 5 projects. And we heard from them all last year. And we're hearing from them all again this year. So we heard from Quantum last month. And I'm pleased to say that we have some early career researchers from MOT4Rivers with us today. And we'll be hearing about how they've progressed over the last 12 to 18 months as we approach halfway point in the programme. So there are a number of ways that you can interact with us, and Finn's just put the slide up here. So you can email us at freshwater@leeds.ac.uk. You can look at the our webpage. You can find all our previous webinars on this webpage, and you can listen to them. And we are on Freshwater Qual 1 on social media. So as I said, we're hearing from each of them this year, each of the projects this year. So coming up in March, we'll be hearing from Quantum, which is really looking at the impact of livestock farming on water quality, followed by Pacific, which is looking at the impact of pollutants on microbial status of rivers, and then LTLSFE in May. So please join us for those if you can. So I'm going to pass over to those that are talking today. So over to you guys. Put it into.
Thank you very much, Pippa.
Perfect.
Can you hear me?
Yeah, can see it and hear you. Thank you.
Excellent. Okay, so I'm Alison Brown from Stirling University. So thank you very much for inviting the MOT4Rivers project to present today. We're looking at monitoring, modelling and mitigating pollution impacts in a changing world. And this is a joint project between the universities of Stirling, Glasgow, Edinburgh, UKCH, the James Hutton Institute, and in collaboration with Scottish Water. So to understand our research vision, we put forward this idea that there's an ecological safe space in which ecosystems can thrive, but this is being limited by chemical stresses, which are forever increasing in this modern world, and also hydrological extremes, also increasing due to climate change. So within the programme, there's 3 work packages. Work package 1, which is a catchment scale monitoring programme, and it's using understanding hydroclimate controls and how they impact pollutant cocktails. And work package 2 is a national scale programme. So this is looking at chemical and biological data across the whole of the UK, historical data, and is trying to understand differential sensitivities of ecosystem communities. And we bring this all together with work package 3, which is a decision support tool for testing interventions and adaptation. So in terms, I'm going to talk about work package 1, which is based in the Almond catchment, a small catchment in Scotland, only 370 kilometres square. It's quite an urban catchment. This is Edinburgh, so it comes to the sea just to the west of Edinburgh. But it's also got quite a lot of other potential pollutants. For example, it's a legacy coal mining area. This dark grey is legacy coal mining, light grey is urban. We also had a lot of oil shale mined in this catchment, and there's a large number of waste heaps, or bings as we call them. Both of these put in a lot of minerals and iron into the river. We've got the M8 and the M9, which run through the catchment. So we've got road runoff going directly into one of the Almond tributaries. There's 8 wastewater plants within the catchment, that's the black dots, and they're putting both treated wastewater and combined sewer overflow input into the river. We've got a lot of urban storm drains, particularly within Livingston here. There's some old rubbish tips. There was also Edinburgh Airport was in the catchment. We've also had open cast mining here and a lot of lime quarries. In terms of other potential diffuse land sources, this shows the land cover of the catchment, and the red spots on here are our 20 monthly measuring points, which I'll be talking about later. So, I mean, in terms of the land cover, there's about 15% urban and suburban cover, about 15% arable colour shown in yellow, about 35% improved grassland at pasture in bright green, about 10% broadleaf, about 10% coniferous, and only about 15% which is sort of natural heather and bog. So in terms of the monitoring program, we're trying to do this on lots of different timescales. So we're developing flow-through systems, which measure using sort of more standard and some more novel sensors, continuous data. We've got our 20 sampling points over the Almond catchment, which we measure monthly, and we're measuring a lot of parameters, nutrients, pharmaceuticals, pesticides, endocrine disruptors, coliforms, metals, greenhouse gases, and various carbon-13 and carbon parameters to help us trace the water. We've got some passive pesticide and pharmaceutical monitoring, so we don't miss anything in our monthly sampling. We're doing invertebrate and eDNA surveys and also some event-based surveys. So primarily today, I'm going to talk about pharmaceuticals, endocrine disruptors, and pesticides. And this plot shows the average over the catchment versus the different chemicals that we've measured. We're trying to measure more, but some of them fortunately don't show up. So the highest concentration of anything we've measured is paracetamol, which comes in about 1 microgram per litre, and followed by other pharmaceuticals, tramadol, ibuprofen, naproxen, diclofenac, trimethoprim, carbamazepine. The highest endocrine disruptor is bisphenol A, and our highest pesticide is imidacloprid. And I'll be talking about both of those in some more detail a bit later. If we look at this in a different way, how they're behaving, we can see that, for example, we've got some very diffuse sources down here. This is atrazine, simazine, and chlorotoluron. These are all general herbicides. We can also see that most of our pharmaceuticals behave as a point source in purple, but also the imidacloprid behaves very much as a point source. And some of our pharmaceuticals, paracetamol, ibuprofen, naproxen, really behave like diffuse sources. So before I go on to show you some of those plots, just to make sure everybody's clear about diffuse and point sources, this plot shows the determinant concentration here in the river flow along the x-axis. And for a point source, as you double the river flow, you typically halve the concentration of whatever you're trying to measure. And then with diffuse sources, they tend to increase in concentration with river flow because rainfall is pushing those more diffuse source pollution into the river. And so I'm going to show a few plots of the almond, looking at the upper almond in light blue, the middle almond in orange, and the lower almond in green. So this is a few of our pharmaceuticals, tramadol here, diclofenac, carbamazepine. I think carbamazepine is quite well known for being very difficult to remove in wastewater treatment. And as maybe you might expect, it behaves almost like a perfect point source, and the other two very similar. But if we compare that with things like paracetamol, ibuprofen, and naproxen, you can say these are primarily behaving like diffuse sources. And our interpretation of this is that as it rains more and you get more water entering the sewer systems, most of it can't get into the treated outflow, and it goes out into the river via the CSO. And these and because these are quite effectively removed by the wastewater treatment, you find they're all ending up in the river. And in fact, if we compare the concentrations in the CSOs compared to the treated outflow, you can see paracetamol being 2,000 to 3,000 times higher, ibuprofen 400 times higher, and naproxen about 24 times higher. I mentioned earlier I would talk a bit about imidacloprid. So imidacloprid is our highest concentration pesticide. It's a neonicotinoid, which is a pesticide that kills bees. And these were banned in 2018 in agriculture, but they're still being used in pet flea treatments or pet parasite treatments. And there's loads of these products on the market. And in fact, one monthly treatment for a dog of imidacloprid is sufficient to kill about 25 million bees. So they're highly toxic and highly toxic to fish and invertebrates. If we look at our plot of our different locations versus our concentration, you can see it's quite easy to get 10 to 20 nanograms per litre, but we've got one location, which is this one up in the in the north of our catchment is getting really quite high concentrations of that. And we actually find even higher concentrations coming out of the wastewater treatment plant there at Winchborough. And you can see from the plot on the right, it is pretty much behaving as a perfect point source. So we think that this high concentrations are pretty much getting in from people washing their pets clothing and their pet bedding. And we're also picking up another neonicotinoid banned pesticide also in this catchment here, clothianidine, at quite high concentrations as well. So if we look at non-neonicotinoids, herbicides, we're also finding those in the catchment, but at very low concentrations, only on average around 3 nanograms per litre. So this one is atrazine, it's the highest that we're finding. And in Scotland, you can't use this on crops in the winter. But if we look at our seasonal distribution, we find that there is some winter use, and this is all coming from this one point here. This is the pink colour, links to this one point here, which has quite high concentrations. So it implies that this is probably coming into the catchment, maybe not from agriculture, could be domestic use, could be from golf courses. This hasn't got any agricultural catchment, this small stream. And we've also found quite high concentrations of atrazine coming in through the wastewater treatment system at this plant here in the middle of the catchment as well. So the other point I'd like to mention is endocrine disruptors. So endocrine disruptors are what's called synthetic estrogens, and they affect the ability of animals to reproduce. The highest concentration we've been finding and the highest occurrence is a compound called bisphenol A at 48% occurrence and reaching 350 nanograms per litre. And bisphenol A is a very ubiquitous chemical. It's used in polycarbonates and epoxies and epoxy coatings, and it's most likely leaching into the environment due to temperature and runoff. The highest place that we find it is just downstream of Edinburgh Airport. We don't know yet if that's a coincidence or not, but you can see that this is reaching quite high concentrations above that of our of the no effect concentration. The other one that we're getting quite high concentrations of and quite high recurrence is the estrogen E1 natural hormone. It's much higher in summer, but it's not perfect point source. So this may not be, sorry, I've changed slides there by accident. It may not be, it may be more a temperature effect, but again, way above our no effect concentration and really covering really most of the catchment. So I'll now hand over to Anna, who's going to talk about the microbiology and the invertebrates.
Okay, thanks, Alison. Hopefully I can control the screen. So hi, I'm Anna Dother. I'm a freshwater ecologist at UKCEH Edinburgh. And for MOT4Rivers, I've been collecting and analysing the river invertebrate samples I'm going to tell you about shortly. But firstly, another aspect of the stream water quality that we've measured in MOT is microbiology. E. coli is used as a proxy for water quality for human health, and the presence of the E. coli itself doesn't necessarily mean that the water is pathogenic, but it's a commonly used proxy for potential negative health effects. And a PhD researcher, Bridget, at Stirling University, has been analysing the microbiology from the monthly water samples and also from 4 wastewater effluent sites monthly. And the map you can see shows a variation in colony forming units between the sites as the months pass. And the colour coding of green, yellow and red on the map is based on the bathing water quality guidelines, just to give you a visual overview of the variation across the catchment. And you can see areas with high and low concentrations. So here values range from the tens to low hundreds in greens in the upper catchment to values of over 140,000 coming from the wastewater effluent. And essentially what you can see here is that in some cases the total coliforms in the river water can be comparable to that measured in the wastewater effluent. And Bridget's work is ongoing to see if proxies can be found to identify the source of the bacteria in the water, whether it's coming from human or agricultural sources. So in the MOT project, we're looking for the ecological response of the exposure to these multiple pressures that Alison's talked about. And one method will be through a eDNA. And we're collecting water samples from 3 locations at each stream site, from a pool, a riffle and a margin. And these represent different areas of the organic material collection in the stream, water mixing and residence time. Some of these samples will be analysed separately so we can understand within channel differences in eDNA patterns, and others will be pooled so that we can quantify the community differences between all the sample locations over time. And outputs from this sampling will be an estimation of the bacterial, invertebrate and fish communities at each sampling location. For the benthic invertebrates, we've followed a 3-minute kick sample with a 1-minute search method in line with regulatory monitoring methods. And this also maintains data similarity to the national data set that's going to be talked about used in Work Package 2. And samples are taken in spring and autumn, and we will have 2 years of data. And these samples are collected at the same locations and same time as the monthly chemistry samples. So how can we quantify an ecological response to the water quality? So on this slide, I'm just comparing 2 sites that are on different ends of the pollution spectrum, the relatively polluted Howburn and the slightly more natural Lynhouse. And generally, we would assume that the more diverse the macroinvertebrate community, it indicates better quality. And just listed here, the number of species in each group and their abundance, looking at the EPT, the river flies, and other taxonomic groups, seas, Coleoptera, mollusks, and Diptera. And you can see that, yeah, for the, oh, someone's moved my slide. Let's go back. Okay, there we go. So yeah, generally for the river flies, there is a higher diversity and abundance in less impacted streams, but the taxa can be quite variable in terms of their species diversity. And looking at the most dominant taxa in these communities by abundance, again, that's very different. The Howburn being dominated by oligochaetes and the Lynhouse by the by Regina. And that's not unexpected because these are different habitats. So the Lynhouse is dominated by mayflies associated with fast flowing and well oxygenated water compared to the Howburn, which is a slower flow and sediment. So our next steps here will be to compare this to a reference community so we can compare like with like in terms of river habitat using the RivPax model. Another way to isolate the effects of water quality is looking at ecological traits that could be sensitive to water quality impacts, either individually or packaged up in a metric here. We've got the SPEAR metric, which is a trait-based metric using a combination of 4 attributes of the invertebrate taxa that indicate sensitivity to pesticides. This combines the traits of sensitivity to the pesticide relative to Daphnia magna, the length of the life cycle of the invertebrates, whether they can use refugia to escape and avoid or recolonize, and the likelihood of exposure during the summer when pesticides are usually applied. And in this plot, I've got the SPEAR score for 8 sites. The score ranges from 0 to 1, which is a ratio of sensitive taxa to all the taxa present. And this is plotted against the total pesticide concentrations from the October sampling season. And in some cases, we see a good relationship where the high metric score, where we've got low pesticide concentrations. And in some cases, like in the Goga 2, there's not a good relationship there. So we've got a low score indicating that it's impacted by pesticides, but we haven't measured pesticides there. So maybe there's something else going on there. Okay, and as well as looking at the species richness and diversity, we can look at the community composition and how the ecological communities at each site are similar or different to each other. And here I'm showing a NMDS plot, which takes into account the taxonomic identity and abundance of all the invertebrates on these 8 sites. And generally, we think that human pressures and impacts leads to the selection of only really highly tolerant taxa and resulting in a homogenized community. And our first look at this data does indeed show that the communities in the less impacted sites, just highlighted here in the circles, appear to be more distinct from impacted sites, highlighted in red, and distinct from each other, compared to the impacted sites, which could be seen to be more homogeneous in their assemblage. So could this invertebrate diversity that we see across the catchment either be protected or restored to support impacted areas to be more resilient to future pressures and contribute to this ecological safe space? So there's the elements that we're looking at in relation to ecological response. So in summary, for achievements and next steps for Water Package 1, we've collected a huge amount of data across a wide range of determinants and locations, and now we've got a good understanding of the impact of the river dynamics and the chemical stresses. Well, some of these sources are linked to CSOs, treated outflows. Some of them imply changes in behaviour from the public are needed. And so next steps for us, we're doing more high resolution data collection, more ecological data measurements and processing of the invertebrates and eDNA samples, and eventually synthesizing the ecological response data along with the physicochemical data to determine the major chemical and hydrological stresses and their interactions into this ecological safe space. So now I'll hand over to Ionut for an update on Work Package 2.
Can you share, okay, thank you. Okay, let's see. Okay, thank you very much. Hello everyone, my name is Ionut. I'm a research assistant at the University of Glasgow, and today I'm going to discuss MOT4Rivers Work Package 2. The overall aim of MOT4 Work Package 2 is to use machine learning methods in order to explore the impacts of chronic chemical exposure to mixtures of pollutants on functionally diverse freshwater biota at the national level across the United Kingdom. Key objectives include analyzing spatio-temporal patterns in pollutant exposure and identifying chemical fingerprints of complex mixtures. The second objective is modeling the effects of biophysical and hydroclimatic drivers on chronic and acute pollutant exposure at the national scale. And the third objective is to quantify the sensitivity of diverse freshwater biota and developing ecological indicators of chemical exposure. While pre-processing the water quality or the chemistry data set, we have faced multiple challenges. The first challenge is data collection. Both the chemistry and the biology were collected from various agencies such as SIPA, EA, or NRW. The data size is another challenge we faced. The size of the data set after filtering for the chemicals of interest is about 1.6 million observations, 2.4 gigabytes in size. And you can see on the right-hand side a map of all the water quality sites across the UK. Another challenge we faced is data quality assurance. Issues faced range from the naming of compounds and even bigger challenges in biology nomenclature. Another challenge is data complexity. The data set is highly complex. It consists of 94 contaminants of different types. The data set are sampled at different frequencies and recorded at multiple UK sites, over 13,000 over a period of 20 years. I have put here an animation of various what we call abacus plots, or where on the y-axis we have the same site number, on the x-axis we have the date, and this plot will just give you an idea of how sparse our data is and what the challenges we face in modeling this. What steps have we taken in pre-processing the chemistry data set? The first step is that we have replaced censored or limit of detect observations using the R package NADA. The second step, we have detected and removed anomalies here defined as very large unlikely values. And we have removed also impossible values here defined as negative values. And only a small proportion of these values have been defined as anomalies. And the first step we have taken is that we have filled in small gaps shorter than 6 months using a simple linear interpolation. And then we have used more complex imputation methods, such as interactive AI approach for gaps larger than 6 months. I have plotted two plots, 2-time series of aluminium and manganese. On the y-axis, we have the log sample values. On the x-axis, we have the data. The original values are represented by the blue points, and the red points represent where we have done the imputing. In both cases, we can see that we have reasonably large gaps where we have filled our data set. After filling in, we can say that the imputing process goes reasonably well. What about the biology? Similar to the QAing of the chemistry data, the biology data has been collated from 3 environmental agencies, SIPA, EA, or NRW, resulting in cohesive data set covering diatoms, invertebrates, macrophysites, with all sites mapped, see image and the data exploration app. This process also faced challenges, including variable data availability, issues in biological nomenclature, the removing of non-target taxon data, harmonizing taxonomic levels, and assessing spatial and temporal data coverage. The CAMEL GB data, encompassing 671 catchment across the UK, see the map on the right-hand side, is also highly valuable as it provides a range of catchment attributes that can serve as covariates in our model. The water quality sites were linked to multiple catchments due to nested catchment structures. However, for modeling purposes, it is more effective to assign each water quality site to a single catchment based on the smallest catchment it falls within, as this is the most hydrological representative. Towards the modeling. We have set up various modeling objectives. These are to develop preliminary models for the water quality chemistry data set. The second objective is to create and classify chemical exposure and ecological sensitivity. And finally, our third objective is to use statistical and deep learning methods to explore relationship between the chemistry and the biology. One of the approaches we're considering at the national scale is to work within each catchment to summarize the chemistry and the biology data before generalizing to a national level. The almond catchment. We have selected the almond catchment as our test case in order to try out our statistical models before generalizing the models to the entire United Kingdom. Traditional analysis has dependent on coincidence measurements in time and space, across stressors, but in our case, this is hugely limiting because, as you can see in the image on the right-hand side, where I have plotted the almond catchment and all the biology and water quality sites scaled by the number of visits. The water quality sites are represented in blue. The biology data is represented by all the other colors. These are not coincident. These are not in the same space, at the same location. So although we have many determinants and thousands of sites across the UK, there are actually a very small number of coincidence measurements. For the almond catchment, originally we have 73 stations and 40 determinants, but once we account for the frequency of monitoring and we're going for more than 120 observations per site and per catchment, we are actually left with just 18 stations and 15 determinants where everything has been, where we have a good set of stations and determinants. And this is even before considering biology. So we are trying to reduce dimensionality of data prior to modeling using a principal component analysis in order to exploit patterns and relationship in the data set. And this is our initial attempt to create an exposure regime. We are considering multiple types of principal component analysis. We can use a standard PCA approach, which is implemented site by site. But given that we have over 14,000 sites, this is computation very demanding. However, we can use a generalized form of PCA in higher dimension. This is also called Tucker decomposition that could be implemented catchment by catchment. And we will have about 614 catchment. So we're going to save a lot of time computationally wise, but the results from an ecological point of view might be harder to interpret. The Tucker decomposition, its input would take a tensor, which is a generalized vector in 3 dimensions, where the 3 dimensions are given by space, that we have multiple sites at multiple locations. The second dimension will be given by the determinants, and the third dimension will be time, given that we have multiple time points where we have monitored all these determinants. And we still have issues then with the non-coincidence of the measurements. Here I have plotted two heat maps of the standard PCA results and the generalized PCA results on the right-hand side. On the y-axis, in both cases, we have the 18 determinants at the Almond catchment. And if we focus on the first plot first, on the x-axis, we have the stations. And if we focus within the box, we see the way we read this is that the parameters oxygen results and the two types of oxygen, these determinants are important contributors, and I have selected the principal component 3 in this case for illustrative purposes. These determinants are important contributors for the vast majority of stations for principal component 3. In general, in the Almond catchment, if you use the standard PCA approach, about 5 principal components, which in our case is defined as a linear combination of determinants, will explain about 70% of the variance. If we move on to the right-hand side plot, I have plotted another heat map, in this case for the Tucker decomposition. And if we focus on the third column for the principal component 3, we can see that we have a PCA loading, a significant PCA loading for, again, for the two oxygen parameters. And we are again shooting for about 70% of explained variance using both methods. And I believe that these two methods can produce similar results in that the two types of oxygen are significant contributors in our case for principal component 3. What about the biology responses? For the biology, the aim is to quantify the sensitivity of diverse freshwater biota and develop ecological indicators of chemical exposures. We are aiming to use standardized taxonomy or literature-based trait assignments, more specifically key metrics such as taxonomic richness, composition, functional richness, and community traits. And we aim to model changes in taxa community traits along chemical gradients, while also accounting for climate and land use effects. So what have we achieved so far, and what are the next steps, and where we are in general? We're trying to obtain a predictive understanding of the ecological and water quality impacts of environmental contamination against a backdrop to changing climatic and hydrological conditions. We have taken a very long time to pre-process the chemistry and the biology data sets. We need to take pragmatic decisions in order to process this reasonably fast. We are trying to maximize the use of available mismatched biological chemical data. We're trying various approaches to deal with the data sparsity, and then to match the biology and the chemistry data sets. We have developed biology metrics to act as a response in our models, and then we'll need to form a regression model in order to link the biology and the chemistry data. And then finally, we're trying to sense check the findings for ecological meaning. Thank you very much for your attention, and I'm going to hand out to control to Mats.
Thank you, Ionut. Hello, my name is Mads Trolborg. I'm from the James Hutton Institute, and I'll just give a brief update on work package 3. So the aim of this work package is to essentially develop a risk model based on a source pathway receptor principle using an approach called Bayesian belief networks. And the aim is to apply this model spatially to the Amman River catchment to identify critical source areas, but also to try and simulate the effect of management and potential mitigation measures on the pollution risk in the catchment. And finally, the aim is also to try and upscale this risk model to national scale application, to basically apply them to these camels catchments that Ionut just talked about. Here we go. So first, what are Bayesian networks? Let me just do like this. And why are we wanting to use them for this project? So they're graphical probabilistic models that represent model variables and the conditional relationships as nodes and errors in a graph. So like this down here, for example, where I've just added a very sort of conceptual source pathway receptor type model. And although it's conceptual, it does actually reflect quite well the structure or general structure of the risk models that we are developing in this project. So we have some pollution source inputs and this pollution can then be transported to the river and potentially result in risk. And how much pollution and how large this risk will be will obviously depend on the magnitude of the source, but also the degree of attenuation that the pollution experienced during the transport from source to river. And all this is then dependent on a number of other factors like hydrology, type of pollutant management, and so on. So Bayesian networks have a number of advantages. So first of all, they explicitly account for uncertainty. They also allow you to integrate quantitative data and existing models with more qualitative information and even sort of expert opinion. So this is very useful when you're trying to model a system where data are scarce and uncertain and where you have knowledge gaps. When you have a Bayesian network, you can then also do these probabilistic inference based on evidence, which is useful for risk analysis and for modeling what-if scenarios. So like, what if we reduce the pollutant load by a certain amount? What does that translate into reduction in risk? And you can even do this in the opposite direction as well, like backwards inference. So you can, oh, there's something missing. You can start from your endpoint and then model what conditions need to be met in order to achieve a low risk. So the risk models that we are developing here basically constitute of a number of submodules as illustrated here, and each submodule, they represent a different type of source. So we have a submodule for sewage treatment works, for example, for septic tanks, for agriculture, and so on. And behind each of these boxes, there's then a more detailed Bayesian network. So the one here just shows the submodule for the sewage treatment work. And the aim of this module is essentially to try and quantify the pharmaceutical load to the river system and the resulting concentration originating from sewage treatment works. And it does this based on a very fairly simple mass balance approach, where the source is determined based on the amount of sewage generated and the prescription rates in the catchment. And the removal processes and attenuation process then include how much is being excreted, the wastewater treatment removal rates, how much is what's breakdown in the environment, and degree of pollution, of dilution, sorry. This slide is just to show some of the required inputs for this module that we've been deriving for the Amman River catchment. So we need some pharmaceutical prescription rates, which are showed up here and that we've derived from NHS data. We also need some river discharge data that we got from the catchment, and we also got some model discharge spatially in the catchment. And we need some physical chemical properties for the various pharmaceutical and compounds that we are trying to model. And this also just shows that there's a lot of variability and uncertainty associated with many of these inputs, not least for the pharmaceutical specific properties like excretion rate and the removal rate in wastewater treatment. So we've done a bit of a sense check of the model behind the sewage treatment work module. So this plot here just shows some simulated versus observed concentrations for 4 different pharmaceuticals at the outlet of the catchment. And for the simulations here, we've actually completely ignored any removal processes. So it's very sort of conservative simulation. So we'd expect our simulations to overestimate the concentrations. And what we just see here is that for some compounds, we actually seem to get quite a good agreement between observed and simulated. There's a nice linear correlation, and our simulated values are generally higher than observed, which can be seen by these slopes of these lines here that are less than 1. In other cases, and other locations, the agreement is not quite as good. It does look like there's a linear trend here, but we massively underestimate the concentration. So there's clearly something unaccounted for here. And in other cases, again, we just get a very poor agreement, and there's clearly some sources and processes not accounted for in the simple module. This is just what the results look like when we're trying to model the risk probabilistically using the Bayesian network, and these results are for diclofenac. So this just shows the probability of exceeding the no effect concentration in the catchment. And we do this monthly, and we do it for every of the 20 monitoring points in the catchment. And what that just shows is there's a spatial variation in the risk, and also a bit of a temporal effect. So it looks like during the summer we have generally higher probability of risk than in the winter, which is due to less dilution. And that's kind of an update, just to summarize the achievements so far and our next steps. So early on in this work package, we completed a literature review on water quality mitigation measures, and we're hoping to use this to inform some of the management notes in our risk models. We are developing, and we're still developing, Bayesian network sub-modules. So the one I showed is probably the most advanced at the moment, but the other modules we have been able to draw on past work, and I've inserted a few links to some previous papers and stuff we've been involved in using Bayesian network for water quality. Then we have spent a lot of time compiling and reviewing pharma-specific inputs like prescription data, accretion rates, removal rates, and so on. And this has been quite time-consuming. The next major step that we've only just started on now is to try and link pollutant mixtures to ecological impact in this ecological safe space. So that's the next one for us to tackle, really. And that's it for me, really. Thank you.